Okay, so let me thank the Cholangio Foundation, Stacy, everybody else, organizers, scientific committee for allowing me to give a talk. And let me tell you, this was not planned that I will talk first, but it fits so amazingly well, and you will see this in a second. So unfortunately, I don't have any disclosures, but that's a different story. So I try to make this understandable both for patients as well as scientists, okay? And it's awkward. We're actually not talking really about the gut microbiome. Actually, we do talk about the gut microbiome, but when we talk about the gut microbiome, we have to talk about something else. By now, you may have noticed I have an accent. I'm German, and I want to introduce you. You may know this. It's a German children's book. It's the story of the little mole, and I found a little video about this book, which basically talks about what we're really talking about. Okay, so this is really the topic. So let's get started. Before we get there, I just want to give you some background what we do at the NIH and how it actually connects to the gut microbiome. So obviously we are very involved in the cholangiocarcinoma treatment. We provide second opinions. We open clinical trials that are actually open to anybody in the United States. Most people don't know this. It's totally free. We treat anybody regardless of insurance because we don't ask for insurance, etc. And as such, I'm just going to give you two examples for studies that we've done. One, Dr. Shea from my group published, where we used one of the immune checkpoint inhibitors, tremelimumab. And the reason why I'm showing you this is not because I'm very proud about the results, because it's really not a good result, but you can see there is one patient that actually has an amazing response. And this is one of our patients, a lady, who up to now is basically without any measurable disease. Now she's been out for 5 or 6 years, and it's not really clear, why we have these outliers. A second study that Dr. Monhe just published, and she's here again, shows you, we do find these outliers. And the hypothesis that I have is that immunotherapy may work in primary liver cancers, but there's just so many unanswered questions that we don't know. And one of them may be actually the connection to the gut microbiome, and that's how we got into the field. So let me start now with the science and why I think that primary liver tumors and cholangiocarcinoma are so important when we talk about the gut microbiome. And that's basically for a very simple anatomic reason. There is what we call the gut-liver axis, which basically means that all the blood that comes from the small and large intestine is drained into the portal vein and goes straight into the liver. And believe me, I'm going to show you some of the data in our mouse studies, but we know this also from patients. This blood, so the blood that goes from the gut to the liver, this is not the same that you get in tests when you draw your blood from a peripheral vein, is actually full of bacterial products, and we know this. And obviously, these bacterial products and downstream have effects on the liver. This is just one study out of some that show that there is an association with clinical response to immunotherapy, actually in patients with immunotherapy. This includes patients with cholangiocarcinoma, and I just want to show you how we do this. We can now use different sequencing techniques to really identify the different commensal bacteria. So these are not bad bacteria. These basically are bacteria, and you will hear this probably more in the first talk, that are actually just living with us. They don't cause a disease. But there appears to be a correlation between the type of bacteria that we harbor or that we have in our gut and the outcome. So let's switch now to the animal studies, how we can actually address and do this. So the first tool we do have is, other than human, we can grow mice in basically completely sterile conditions. These are called the gnotobiotic mice or germ-free mice. And the other option that we have is that we treat mice with an antibiotic cocktail to pretty much deplete all the bacteria that you can see here within a very short period of time. So a number of years, we did this in a transgenic mouse model. The first part I'm going to show you briefly is about HCC, and then I will talk about cholangiocarcinoma. And what we observed in this mouse model is that these mice are genetically engineered to develop liver tumors. But if we treat them with antibiotics, the tumors disappeared. They wouldn't grow. And I see enough patients that I know it's not that easy that you can just treat a patient with antibiotics. So we got really, really interested in how this works. And we did this the same that we do in our patients. We basically profiled the immune system and the immune cells in the liver. And we noticed that there was a specific accumulation of cells in the liver, so-called NKT cells, in all those mice that were treated with this antibiotic cocktail. NKT cells are very specific immune cell. I don't want to bore you with all those details. They are kind of in the middle between innate cells and T cell. They are very abundant in the mouse liver. They recognize lipids, and we can easily identify them using a specific antibody for the V-alpha chain of the T cell receptor. And finally, most importantly, they express CXCR6. So we looked in our mice in the livers, and you can basically see on this slide that these NKT cells, which we can stain using a CD1D tetramer, are CXCR6 positive. And if you just look at CXCR6 positive cells in antibiotic-treated mice, that's what you see on the lower right of the slide. I'm not going to use my pointer because I think it's pointless to use the pointer because I don't know who can see it online or here, so I'm just trying to explain where I am. So anyhow, so the antibiotic treatment leads to the accumulation of CXCR6 positive cells. CXCR6 gave us basically a key that this may be related to CXCL19. And indeed, we do find in the liver a higher expression of this chemokine CXCL19 in mice that were treated with antibiotics. And this chemokine was synthesized by so-called liver sinusoid endothelial cells, which are cells that are basically within the portal vein in the liver and are really the first barrier that the blood hits when it goes into the liver. We can stain this and show this. So what I've shown you so far here is that in the context of antibiotic treatment, we kill all the bacteria. This leads to an accumulation or an increase of this chemokine in the liver. This chemokine makes the NKT cells migrate into the liver and then prevents tumor growth. Now the question is still, how does the gut microbiome and the CXCL16 connect? And we basically found out that this is related or that this is controlled by bile acids, which are under normal circumstances released by the liver into the small intestine and then actually metabolized by these bacteria that we have in the gut from primary bile acids into secondary bile acids. And if we deplete mice of these bacteria using this antibiotic cocktail, basically we are no longer able to metabolize primary bile acids into secondary bile acids. So if this is really the case, we can actually modify this in mice, and we can also look at the bile acids in these mice. And indeed, we did find an increase of primary bile acids in the system when we treated mice with antibiotics, and at the same time, a reduction of secondary bile acids, which you can see on the right, basically just because we eliminated these bacteria in the gut that can metabolize primary into secondary bile acids. Now, here we show you now that indeed we can use bile acids to treat tumors in this system. So the experimental setup is shown on the right. We treat mice with an antibiotic, in this case vancomycin, to deplete the bacteria. Then we challenge these mice with a tumor that grows in the liver, and we treat the mice with a secondary bile acid, and you can see on the left the outcome. Antibiotic treatment, I already shown you this, reduces the tumor growth in the liver, and then we can basically reverse this by adding secondary bile acids. So here, this is basically the first proof that indeed using a mechanism by bile acids, we can modify immune cells, which I don't show you here. We studied this in parallel, but we can actually see a difference in the growth of tumors. Now, this doesn't show you yet that actually specific bacteria can do this. So we went one step further. We looked at the bacteria in more detail, and I told you there is one specific bacteria that actually codes for a specific protein or alpha hydroxylase, which you need to metabolize primary into secondary bile acids. And what we can do is we can actually bring this bacteria back into these mice, so we can colonize the bacteria that's under normal conditions in this individual, and thereby change the number of NKT cells, which you can see on the right side. But the most important thing is the tumor outcome, which you can find here on this experiment, where again we treated mice with antibiotics, challenged them with the tumor, and then selectively recolonized them with Clostridium scindens. And you can see that indeed that just adding one specific bacteria into your commensal bacteria into your gut controls tumor growth. Obviously, in this scenario, what you see here, there is more tumor growth. So this is obviously something that you would not want. But the reason why this is so important, because we basically can understand the mechanism now and can better understand, can potentially understand how we can also use this therapeutically. So let me summarize this part. I've shown you that under normal circumstances, you have bile acids that go into the small intestine produced by the liver. Depending on the presence or absence of specific bacteria, Clostridium scindens, you have more or less primary and secondary bile acids. They go back into the liver through the portal vein, where they induce the release of a chemokine, CXCL16, which in turn leads to the accumulation of CXCL6 positive NKT cells, and these NKT cells control tumor growth. Now let's go to cholangiocarcinoma. Before we go there, I just want to show you, we're actually testing this also in patients, where we treat now patients with vancomycin to see if these observations that we made in a completely artificial preclinical mouse model also apply to patients. So now let's switch to biliary cancer. And here we decided to initially start looking at this particular idea that there are certain risk factors. Patients with primary sclerosing cholangitis, inflammatory bowel disease are at risk for developing a cholangiocarcinoma. And it was known that these individuals actually have what we call dysbiosis in their stool. So that is basically the composition of normal or commensal bacteria is different in a healthy control versus patients with a PSC or inflammatory bowel disease. And you can see this on the left and also on the right of this slide, where we compare healthy controls, which are circled in red. And you can see that the different composition of the different bacteria is very different to patients with PSC or ulcerative colitis or Crohn's disease, indicating that there may actually be a relationship indeed between the gut and the outcome. So we use two different mouse models that we have in the lab to induce cholangiocarcinoma. I don't want to go into too much into details, how we do this, but we can transduce these oncogenes into the liver so these mice will develop tumors. And we also use two different mouse models which somewhat mimic the situation of an individual with a PSC, where we get inflammatory responses and fibrosis in those livers. And if we do this and look at the gut of these mice, imagine, so remember, we only actually do something to the liver. We don't do anything to the gut, primarily to these mice. We see that these mice develop severe colitis. You can see on the H&E stain, that is the red or stain on the lower left, that the membrane of the cecum is no longer intact. We see a reduction of tight junction proteins such as occludin. We find a change, an increase in pro-inflammatory cytokines. We can actually find bacteria in the portal vein, as well as if we look in patients, we can find signs of bacteria appearing in those cirrhotic patients. If we look deeper into these livers, we find that there are specific changes in the immune environment of these mice. There's an accumulation of polymorphonuclear MDSCs, which you can see on the upper left. MDSCs accumulate in mice with PSC in this model, and we can further subdivide this and focus on the different subsets of MDSCs and see that this is primarily the polymorphonuclear MDSCs. This is not limited to one mouse model. We can find this in a second mouse model shown on the lower left. And then, interestingly, if we treat these mice again with the antibiotics, and remember with this we really perturbate, completely change the gut bacteria, we don't see this increase anymore. And now we can even do this more specifically by using specific bacteria that would not just eliminate all bacteria, but only part of the bacteria. And you see, if we do this, in this case with neomycin, you see a clear reduction of these mononuclear cells. As well, if we do the experiment in a little bit more sophisticated way, by using what we call an FMT. So in this case, we actually transfer the stool samples from one mouse to another mouse, and the initial mouse was treated with different antibiotics. We see a difference in the cells in the cell subset. These cells again express specific cytokines. In this case, CXCL1, chemokine CXCL1 on their surface. And we can even understand now how the cells are regulated. They are basically regulated through a TLR4-dependent mechanism, which is shown on the lower left. We can block this interaction between TLR4 and CXCL1 to change the accumulation of MDSCs in the liver. Now, what I've shown you so far is this. There was no cancer. All I've shown you that in the context of liver cirrhosis, there is a leaky gut. We believe that it's LPS that induces this expression of CXCL1 in hepatocytes, which leads to the accumulation of CXCR2 positive polymorphonuclear MDSCs. And this appears to be dependent on the TLR4 expression on hepatocytes on one side and gram-negative bacteria in the gut on the other side. So in the last 2 minutes, my time is over, I'm almost done. I'm going to show you now how this actually relates to a cancer model, because so far I only talked about liver cirrhosis. So we combine the two models. We combine the model of PSC and colitis, and you can see that indeed those mice develop more tumors in different settings, including colitis. But now if we actually block this interaction, we either block the interaction of CXCL1 or we treat these mice again with antibiotics. You can see that indeed neomycin treatment reduces the number of tumors, again in an FMT study, so there's no direct effect of the antibiotic. It's really only the stool transfer that we do that has an effect on tumor growth. And again, this is TLR4 mediated, similar to what I have shown you before. So let me summarize, and the work was done by Shanfei Zhang, a former postdoc in my lab, and this work was also funded actually and supported by the Cholangio Foundation. So under normal conditions, in case there is a change in the liver, such as fibrosis, we start to see a change in the microbiota composition. This may be induced by change in bile acids in the system. The change of these different bacteria that are, again, normal bacteria, causes a leaky gut, which means the bacteria can transmigrate easier into the gut epithelium. They go through the portal vein into the liver, and through a TLR4 mediated mechanism, lead to the accumulation of these MDSCs, which suppress immune responses, and thereby ultimately suppress the function of effector cells that can control tumor growth. So with this, I would just briefly like to close and tell you, so the microbiota, I just showed you one aspect, but we do know that there is obviously many microbiota-dependent regulation of cancer development, progression, and treatment. So this can be, I've shown you the idea of liver fibrosis causing dysbiosis in the immune response, but there's many different ways how you can change the gut microbiome just by diet, lifestyle, etc. By treatment with antibiotics, xenobiotics. We will talk here about this a little bit later. I told you about the fecal transplantation. I've shown you that change of the microbiota can change the microenvironment and immune cells in the tumor environment, especially in the liver. And then this obviously has effects on cancer progression, but it also has effects on treatment responses that you will hear later, and comorbidity. And with this, I would like to close, thank my group and the collaborators that helped us do these studies.
Thanks very much, Dr. Greten. We'll have Dr. Sepich-Poor come up.
So sorry for those technical difficulties. All right, so just quick disclosure, during my PhD, I co-founded a company called Micronoma and have filed various intellectual property on some of the information presented today. Many of us are here because of family members or friends or ourselves who have experienced the diagnosis of cancer. I am in medicine because of one such story with my grandmother. This is my grandmother pictured in 2011 at a Phillies game. She loved baseball, and she's pictured here with my dad behind home plate. This was my grandmother 1 year later, one day after her pancreatic cancer diagnosis. Minimal symptoms, some diarrhea, bloating, almost nothing else. It was a shock to our family, and it was a shock to me. I was a freshman in college at the time, about how quickly this disease progressed. She had 33 days from diagnosis to death. They trialed her on very intensive therapy, which unfortunately failed within 3 weeks. And at that point, the side effects were so severe that she decided it would be better to live with the cancer. I decided to go into medicine in part to try to better understand what new diagnostics and therapies may be able to help patients of all cancer types, like my grandmother. I was surprised that in the case of pancreatic cancer, despite the massive genomic revolution that's happened in the last 50 years, the 5-year rate of survival has increased from about 3% to about 10%. As a second-year medical student, I was then shocked to see an article published in the journal called Science, where researchers between the Weizmann and Broad Institutes identified intratumoral bacteria in 3/4 or 76% of pancreatic cancer patients. Notably, this was a completely accidental finding. They had actually been studying chemotherapy resistance, and they noticed that some of their cell cultures were contaminated with a bacteria called mycoplasma. But those contaminated cell cultures were becoming resistant to gemcitabine therapy. Curious, they explored this a little bit in more detail and found that there was an enzyme shared by more than 300 species of bacteria, many of which are common to the gut, that can enzymatically degrade gemcitabine to a non-functional byproduct. And in fact, they could cultivate bacteria from these patients' tumors and then put them in new cell cultures and show that these new cells had resistance to gemcitabine therapy, meaning that patients, simply by bacteria inside their tumors, could become resistant to treatment. A paper 2 years later, published by MD Anderson, showed that just using intratumoral bacteria in pancreatic cancer could differentiate survival by 9 years, a median. This was astonishing to me because, after all, isn't cancer supposed to be sterile? Now, I posed on the previous slide, maybe oncologists should be talking more to microbiologists, which is the theme of my talk. Now, when I studied my PhD, I started in a lab that focuses on the microbiome. I had no experience with the microbiome prior to starting in the lab. But as I quickly came to learn, most of life is microbial. If you plot the history of the world on a 12-hour clock, microbes came in around hour 1:30 in the morning. Humans came around the last 2 seconds of that 12-hour clock. If you look at the genetic diversity that's present in the world, it is mostly bacterial. Bacteria can survive in almost any climate that we look for them, and yet, again, tumors were supposed to be sterile, or at least the hallmarks of cancer, which have been collectively cited 95,000 times since their publication in 2000 and 2011, have nothing to do with microbes. Are we sure, with certainty, that these are actually sterile? This is what I focus my PhD on, and I think it's very relevant to cancer diagnostics and, of course, therapeutics, as I've already described in the case of pancreatic cancer. So we started off by looking at the largest cancer genome database in existence, the Cancer Genome Atlas, which includes cholangiocarcinoma. This includes over 10,000 patients that were sequenced, had their tumors sequenced and blood sequenced and adjacent normal tissue sequenced from 2006 to 2016. We profiled what everyone else thought was, quote, trash. What I mean by that is all the DNA and RNA in these studies that did not map to human reference genomes. And instead, using that information, we compared it against a very large database of 60,000 characterized microbial genomes. We applied in silico decontamination methods, removing up to 92% of the data. And then we applied machine learning to test the basic question, are cancer microbiomes different from each other? And of course, for the future, there's all this human information that can be integrated with it. Now, this was the computational equivalent of looking for needles in a haystack, where there are more things of hay than there are stars in the Milky Way, actually 60 times as many stars in the Milky Way. It was a massive computational effort. But what was really surprising is that no cancer type was sterile, as in every single cancer type had a non-zero number of microbial reads associated with it. And this was true whether you looked at the total, as in anything that was mapped to microbial, or things that gave you sufficient resolution to go down to the genus level. Now, this is interesting because another effort which looked at healthy individuals and many different tissues from healthy individuals, which was published in 2018, found roughly the same proportion, I should say slightly less proportion of microbial reads in their own data. And so it made sense that individuals with cancer may have a larger amount of microbial reads. In the following slides, I'm going to present some machine learning data. Effectively, the basic model we used is 70% for training, 30% holdout for testing, upon which you use to evaluate how well your model learned the information. Now, this is a heat map. The hotter the heat map, the better the machine learning performance. In subfigure A, we are performing a one cancer type versus all others. This means that we are looking at samples from an individual cancer type versus 31 other cancer types. And we're trying to ask the basic question, can you distinguish a cancer type solely using non-human microbial DNA or RNA? And as you can see, the heat map is very hot. These are all areas in the ROC curve where 1 is the maximum, and they are very, very close to 1. In fact, the average is like 0.97. If you look at distinguishing between tumor tissue versus adjacent normal tissue, it is also very hot in the sense that you can distinguish between those just using microbial information. Now you can zoom into any one of these boxes, and this reveals an ROC curve with a respective confusion matrix. Now, I really love Tim's talk because it presented the idea that bacteria may find their way into circulation. This is curious and actually has been documented since the 1970s. This is a 1977 New England Journal of Medicine paper where they related that patients who had Streptococcus bovis bacteremias later had subsequent colorectal cancer diagnoses even without any overt symptoms of colorectal cancer. This suggested that maybe blood samples, and specifically microbial DNA within blood samples, may be indicative of the presence or type of cancer. Fortunately, we had 20 cancer types in our data set with this information. And again, as you can see, we are performing a one cancer type versus all others, just using microbial DNA found in normal blood from these cancer patients across 20 cancer types. This is very surprising, to say the least. The fact that we could distinguish the type of cancer just using non-human information. We then validated this. We collected 169 plasma samples, 100 of which came from 3 different cancer types: lung cancer, prostate cancer, and melanoma, and 69 of which were serotyped HIV negative non-cancer controls. And what we found is that, I'm presenting one of the data points here, in lung cancer, we could distinguish between individuals with lung cancer and non-cancer individuals with an AUC of 97%, which is phenomenal, again, using non-human microbial DNA and plasma. And I'm not suggesting these microbes are alive. This is simply cell-free DNA that is being shed into circulation from some source, either from the gut or potentially the tumor. Now, I'm going to present some unpublished data where we took another paper that was previously published by Johns Hopkins, Victor Velculescu's group, in 2019, where they were looking at host-based fragmentomic methods for early-stage cancer diagnosis. We took their retrospective data and applied our microbial mining methods to their data. Now, what was great about this is that all the patients were treatment naive, they had many different cancer types, and they were mostly early stage. They also include 25 bile duct cancer patients in this cohort. We then tried to distinguish all cancer types versus healthy, and again, very surprisingly, found that solely using cell-free microbial DNA, we could very accurately, with high sensitivity and specificity, distinguish healthy individuals from those with cancer. And in fact, in bile duct cancer, the AUCs approached 1. Now, our previous studies mostly focused on the contribution of bacteria and viruses in tumors. What has been understudied is the relevance of fungi. Now, fungi are still an important part of the microbiome, and in some cases, they can actually symbiotically work with bacteria, for example, to form biofilms, such that bacteria can actually adhere to local fungi, which can be involved in disease progression or even therapeutic resistance. So we worked with collaborators at the Weizmann Institute, this is also unpublished, in order to see how useful fungi would be if we added it to our models to improve how well we could distinguish between cancer types, again, just using non-human microbial information. And as you can see in multiple cohorts, whether looking at tumor tissue or in blood, there's a synergistic effect such that fungi are contributing novel information that's not previously seen just by bacteria alone. Now, again, returning to this Hopkins validation cohort, if we look at fungi by themselves, the performance is not that great. It's an AUC of about 70%. But if you add bacteria and fungi together, the performance for distinguishing stage 1 cancers versus healthy controls approaches roughly 95% area under the curve, which is state of the art. This is about as good as it's been seen in this data set. And it's consistent across all stages. Now, I've been showing you lots of data, right? And we all like pictures, perhaps a little bit more. So our collaborators at the Weizmann in a separate paper did the hard work of imaging bacteria and tumors. Now, what you can see on the left is H&E stains, also looking for LPS, which Tim talked a little bit about, as well as looking for 16S rRNA, which is a ribosomal subunit found in all bacteria. If you look at the percent bacterial composition, it approaches that of PD-1 positive cells. Now, I realize this is not a lot, but I don't think anyone in the audience would argue that PD-1 positive cells, even though they're lowly abundant, are not useful for cancer therapy. In fact, a large backbone of immunotherapy is based off of PD-1 positive cells in the tumor microenvironment. Most surprisingly, this group at the Weizmann, led by Ravid Straussman's lab and others, have actually characterized that many of these bacteria are intracellular. People are still trying to figure out exactly how this happens. We don't fully understand, but we do know that they're metabolically active, and that in certain cases, they can degrade xenobiotics, which are compounds that humans can't metabolically process, while inside the tumor. You can also find fungi if you stain histologically. It is more difficult to identify fungi. They're less abundant, and the stains are a little bit more difficult to work with. But again, we are able to find bacteria and fungi in tumors. So to summarize, maybe we shouldn't be treating all these cancer types as sterile, and maybe there's useful information we can gain from this. I want to acknowledge the Microbiota team, and since I have about I'll just conclude here, I have 1 minute left. I don't think we should be treating tumors as sterile entities. Now, this unfortunately opens up a Pandora's box of exactly how we should treat tumors. Giving antibiotics may not always be the best interest of the patient because it often abolishes responses to immunotherapy, which may be useful, especially in late-stage disease. It appears microbial DNA can be detected consistently in blood and plasma. We do not know what the sources are as of yet. It appears that this is a multi-domain ecology, as in it comprises bacteria, viruses, fungi, very likely phages, which haven't been well characterized to date, and lots of other things. And so my challenge for us as a community is maybe we should work with our oncologists to consider how we can utilize this information for better diagnoses and therapies for everyone with cancer, including those with cholangiocarcinoma. Thank you so much.
That's a fantastic talk, both of you. It's certainly my honor to be the third speaker in this group, and my talk is entitled The Role of the Microbiome in Predicting Immunotherapy Response. And I added a clinician's overview of emerging data, since I am most definitely a clinician and not a lab-based researcher and not even a microbiome researcher, though I am a clinical researcher in immunotherapy, and so this is a topic that's extremely important in the immunotherapy field as it emerges, and I'm very glad for the opportunity for this impetus to learn quite a bit more about the topic. So thank you to the organizers for the invitation. Here are my disclosures. And again, at the bottom, I am not a microbiome researcher, but I've learned quite a bit in preparing for this talk. So here's the quick outline that I'll follow. First, a quick background, again, from the clinician's perspective. And then I'll spend the most of the talk reviewing the emerging evidence for microbiome impact on immune checkpoint inhibitor, or ICI is the acronym I'll use throughout, treatment outcomes, both efficacy and toxicity-wise. And then touch briefly upon some of the emerging data that suggest potential for therapeutic implications and future directions. So again, just a brief overview about the microbiome, which both Greg and Tim have touched upon before this much more thoroughly. The human microbiome refers to commensal microbes, including bacteria, viruses, fungi, and archaea that are other prokaryotic single-cell organisms, which inhabit the human body throughout, and we're just beginning to learn their roles, both as commensals within our body and also intratumorally as a pathogen. This is an inordinate number of cells in our body, 4 times 10 to the 13th on average, with up to 1,000 different species and vastly more subspecies. And these encompass over 100 times more genes than the human genome itself, so an enormous amount of data to sift through. Our initial colonization occurs at birth from maternal exposure, and our microbiomes individually are highly heterogeneous, influenced by diet, medications, geography, exercise, other lifestyle factors. And you can see on the right this figure from a review by Gilbert et al. In Science a couple years ago, just showing how different two healthy individuals' microbiomes might be in different parts of their body. So again, heterogeneity is the rule here, even in the normal setting. And the GI tract is the most densely populated region of the microbiome. So what is the relationship between microbiome and cancer? Well, there are a few microbes that are directly carcinogenic and have been recognized as carcinogens by the International Association of Cancer Registries. But there are also a host of commensal microbes that we're really talking about today that can impact carcinogenesis in varying ways without being the direct cause of cancer. And these include both good bacteria or microbiomes, which can be immune stimulatory or protective, the second bullet there, and also those that can be complicit or promote cancer in various ways, either by immunomodulation or through their metabolites, for example. And then acknowledging the morass of bacteria or microbes that we're talking about, others may be purely bystanders and confound our data. So it's certainly complicated. So how is the microbiome measured? Well, we can measure the presence of an individual taxa or class or grouping of microbes, the abundance of those groups or taxa, the richness, the number of individual taxa present, the diversity, which includes richness and abundance. Both alpha diversity is within sample diversity measured with a lot of different indices, or beta diversity, which is cross-sample diversity. We can measure these over time in response to different interventions such as therapy, as well as, again, a huge host of other surrogates such as metabolites. Shown on the right is something that Tim showed in his talk as well, the bacterial taxonomy. Goes from phylum to class to order down to species. And the 3 measures that we'll talk about most in the data I'm going to show you today are abundance, richness, and diversity. And I couldn't resist showing this mnemonic in case there were any of you like me who had this floating in their mind. Dear King Philip came over for good soup, which helps us remember these, and also good for those of you like me who have a middle schooler at home learning this. So I think this is one of the early landmark papers looking at the impact of microbiome on efficacy of immune checkpoint inhibitors. This is in a melanoma cohort where the majority of the data begins. A small cohort of responders and non-responders to anti-PD-1 therapy had their baseline and serial microbiome sampled, and we can see on the right the graph with the blue and the red bars that they distinguish themselves by responders and non-responders according to the most prevalent bacterial taxa present. Responders had higher abundance of specific groupings called Firmicutes, Clostridiales, and Ruminococcaceae, based on the analysis. I should add that these are all sort of overlapping categories. Firmicutes are phyla, Clostridiales are orders, and Ruminococcaceae are family, and so these are somewhat overlapping Venn diagrams within the cluster analyses done. But the point taken here is that the responders and non-responders had different microbiomes. There was also in this study higher alpha diversity or intra-sample diversity, number and abundance of microbiota in the responders. And that was associated with progression-free survival. So a very interesting clue of this impact clinically on immune checkpoint response. Now looking at different tumor types, another landmark paper by Rowdy et al. Here and looking at predominantly non-small cell lung cancer, NSCLC, and renal cell or RCC cohort, also showed that richness or measurement of diversity was associated with longer progression-free survival in the chart on the left. And again, they also showed in the green and red bars that the relative abundance of specific taxa was associated with response status, responder or non-responder. They again showed that the phylum Firmicutes was enriched in responders and specifically called out a species, Ankermansia mucinophilia, which was significantly associated with response in the non-small cell lung cancer and RCC patients in this, again, small study. So what do we know in hepatobiliary cancers? So a recent paper by Mao et al. In JITC looked at 65 patients, a combination about half each of hepatocellular carcinoma and biliary tract cancer from Peking Union Medical College. And they showed again that they could, and what they did was they dichotomized these patients according to clinical benefit response, which was defined rather broadly as partial response, complete response, or stable disease for at least 6 months, versus those with no clinical benefit, or NCB, which were defined as primary progressors or stable disease less than 6 months. And again, those sorted into about half and half. And looking at the CBR versus the NCB groups, we could see that there were differentially abundant taxa according to response status. Looking further, they could see differences in abundance at both the phylum and order and across levels of taxa for the responders versus the no clinical response groups. There was enrichment of bacteroidetes and Firmicutes phyla in the responders versus the proteobacteria were enriched in the non-response. HCC and BTC were relatively similar to one another with slightly higher enrichment for bacteroides in the biliary tract cancer cohort. And reminiscent of the melanoma data, they did see that the Ruminococcaceae family was enriched or more abundant in the responder group. So perhaps an early recurring theme there. And looking at the functional question here, an interesting point of this paper is they looked at the genes and metabolic pathways associated with the taxa that were upregulated, or enriched in the CBR versus non-clinical benefit groups, and found that there were different metabolic pathways. Those associated with energy metabolism were enriched in the clinical benefit group versus those involved in amino acid metabolism, which were enriched in the no clinical benefit group. Now, changing directions a little, how does the microbiome impact on toxicity? This was shown here is a paper by Andrews et al. Published in Nature Medicine earlier this year. Looked at a retrospective and prospective cohort of patients with melanoma treated with immune checkpoint inhibitor therapy and took serial samples before and during treatment, and then looked at patients according to their immune-related adverse events, and those with severe grade 3 or higher were studied for this analysis. And what they showed is that, again, if sorting according to the presence of severe immune reaction, they could also call out specific taxa that were more likely to be present in those with immune adverse events versus those without severe immune adverse events. The blue and green bar charts, again, show these sortings. And they didn't show any difference with diversity, but they did call it specific taxa, including Bacteroides intestinalis was higher in patients with severe immune-related adverse events. And then getting into the mechanism, they showed that a particular metabolite, or excuse me, a cytokine elaborated by certain bacterial groups, IL-1 beta, was higher in patients with immune-related adverse events of high grade than those that were not. And again, and then they studied this in mouse models and showed a mechanistic rationale for this finding, that particular taxa of the microbiome could elicit this cytokine response and then promote the immune-related toxicity through that, mediate the immune-related toxicity through its cytokine impact. And with respect to hepatobiliary cancers, there's also a hint that potential microbiome may impact on immune checkpoint inhibitor toxicity as well. From the Mao paper I showed you earlier with respect to efficacy, they found that patients with high grades of diarrhea were more likely to have decreased baseline gut microbiome diversity and enrichment for a particular species, Prevotellomycelia timonensis. So just to summarize what we have here with this recurring theme, I'd say this is a recurring theme of dysbiosis, and again, hearkening to Tim's talk in particular, where we see that relative abundances of specific gut microbial taxa can be correlated with immune checkpoint inhibitor efficacy outcomes across a variety of tumor type cohorts. I've shown you solid tumor data here from melanoma, non-small cell lung cancer, renal cell, urothelial, et cetera, shown here. But there's significant heterogeneity between studies, and the bacteria or particular taxa that are enriched in one do not always recapitulate in another. Now, some of these studies show that higher alpha diversity is related to improved outcomes in melanoma, renal cell, and non-small cell lung cancer in particular. But in some follow studies, including in melanoma, with larger studies, these have not been substantiated. In fact, alpha diversity was not associated. So again, this still is an unfolding story without a conclusion. But I think all of the data so far do support that favorable and unfavorable microbiota profiles can impact immune checkpoint inhibitor response and toxicity. So what are the therapeutic implications? Can we use the microbiome as a predictive biomarker, particularly for immune checkpoint inhibitor response? Can the microbiome be modified therapeutically? And the obvious questions that come to mind are diet and supplements, antibiotics, and fecal transplant, and both Tim and Greg have touched upon these as well in preclinical models in particular. And so it's taking the first of these questions. I'm going to quickly go through these because there's not a lot of data, but I think these are fascinating future directions. So can the microbiome be tested as a biomarker? I think right now this is premature and definitely not ready for the clinic, because the data we have are really small retrospective cohorts with significant heterogeneity and inconsistency in results as well as methodology, and there are many confounding factors. But I do think that what we are learning from this data so far, as preliminary though it may be, is that microbiome biomarkers, including potentially the abundance of specific taxa or patterns, may warrant further, or definitely warrant further prospective study in randomized cohorts. Now, this slide I think just shows is a cautionary note from the Andrews paper, a follow-up larger study in melanoma that showed that if one looks at the top hits of taxa associated with positive response or clinical benefit in the study shown here by Andrews, and compares it to 5 other melanoma cohorts that have been studied in the past, there's really a lot of heterogeneity where concordance is rare. I think the only bacteria taxa that showed concordant results in terms of benefit versus negative outcome were the bottom one, the ruminococcus, which was blue in most of the studies, though there's still an outlier so it was favorable in the study shown in blue where it was associated with positive benefit, yet there was still one study where it was called out as a negative. And so again, this is a lot of heterogeneous results. It depends a lot on the details of exactly which species are being measured and how. So quickly, can diet and supplements favorably modulate gut microbiome? Now, this was a recent paper that was published in Science in December of a melanoma patient cohort treated with immune checkpoint inhibitors that underwent fecal microbiome profiling at baseline and serially on treatment. And the authors tracked the dietary fiber intake as well as the use of commercial probiotic supplements in this cohort. And about 30% of patients had taken probiotics, and about 30% had what we would define as a high fiber diet greater than or equal to 20 grams per day. And then they examined this cohort for clinical outcomes. So this slide shows simply the survival outcomes of patients who did or did not take probiotics. And though the curves are pretty close, we can see the top darker curve is patients that did not take probiotics had a slightly more favorable survival probability, acknowledging again all the potential covariates, but certainly a sobering reminder to be cautious about supplements. They then followed up this result in the human population with a mouse model, taking patient fecal matter and injecting it into a mouse, and then gavaging that mouse with several different kinds of probiotic, either a bifidobacterium-based or lactobacillus-based probiotic, and then treating with immune checkpoint inhibitor for a melanoma model of tumor. And one can see in the light purple and the medium purple color that the tumor size was greatest in the two cohorts of mice that received the probiotic. So it promoted tumor growth in this mouse model to take one of these two types of probiotic, whereas the controls without probiotic had the slowest tumor growth. And the putative mechanism for this finding was elicited by immune profiling by flow cytometry of tumor microbiome, or tumor immune cells, which showed reduced interferon gamma positive or effector T cell profile in the tumors of the probiotic-treated mice. So again, a hypothesis that perhaps the probiotics were causing some immune suppressive effect in the microenvironment. Now, in that same cohort, the intake of dietary fiber in the dark curve on top was associated with improved survival probability, again in a small cohort, and so was a more favorable outcome and was also recapitulated in their mouse model. Another huge topic in one slide here, what is the impact of antibiotics? We've already heard a bit of mixed response, are antibiotics good or antibiotics bad? There are several retrospective and observational studies that have shown poorer outcomes on immune checkpoint inhibitors in patients who required antibiotics. But as Tim showed us, there's also potential for benefit. And one study I wanted to draw your attention to from Fessas et al. In liver cancer last year was a large international cohort of 450 HCC patients, which showed no detriment in outcomes on immune checkpoint-based inhibitor-based therapy with early antibiotic exposure, as shown in those survival curves. With the red being the patients that received antibiotics early on in their course. And in fact, the PFS trend favored patients who had received antibiotics. So again, this is a message that we don't really know the impact in the clinic, and it's probably much more complicated than just a simple yes or no. So I'll skip here, and just to go to the summary of therapeutic implications, second to last slide. No current role for microbiome biomarker testing in the clinic yet. I think Greg's data was fascinating about the potential role as a diagnostic, which may be even further ahead than as a predictive biomarker. Certainly prospective studies are warranted to validate the microbiome-based biomarkers in the future for immune checkpoint inhibitor response or toxicity. No current role for supplemental probiotics, and just a warning note that these could be detrimental depending on context and type. A high-fiber diet continues to show no harm and potential for benefit across a lot of health contexts, so I think that we can favor that. And the impact of antibiotics is really unclear on immune checkpoint inhibitor response, and antibiotics should be used as clinically indicated for infectious reasons. So future directions. I think that the TOPAZ-1 regimen, which we'll hear about later today from Dr. O, has established a new role for immune checkpoint inhibitor in the treatment of cholangiocarcinoma. And we do need prospective studies of relevant microbiome populations in cholangiocarcinoma to characterize the microbiome and what is eubiosis or what is dysbiosis, and how do they relate to immune checkpoint inhibitor outcomes specific to the cholangiocarcinoma context. And there's much work ahead, but enormous potential for benefit. So thank you so much for your attention.
Thanks very much, Professor Kelley. I think, Stacy, we're going to help us with, I think we have about 10 minutes for questions. Will you please come up? I think we have some mics that are back in the audience, so please come up. Dr. Smoot, is that right?
Yep. Hi, thanks, Rory Smoot, Mayo Clinic. Greg, very interesting findings. How stable are the sequencing findings? For example, if you take the same patient and test them multiple times, do you see stability of the sequencing findings, or does it modulate? And any effects of treatment, do you know, people on therapy, off therapy?
It's a great question. With respect to tumors, I will note that it's very difficult, unfortunately, to get longitudinal samples. And so it has not been possible to date in order to look at longitudinal changes before and after response to therapy, especially if the therapy is effective. There usually isn't a lot of tissue to work with afterwards. In terms of blood and plasma samples, it is an active direction of research. We have noted some preliminary data where there are distinct changes in what I'll call the plasma microbiome after therapy. But the one tricky variable here is that every type of therapy may elicit a different change. What I mean by that is, for example, there are well-established, say, colitis side effects of immunotherapies for certain kinds of immunotherapies, which may increase the amount of translocation of bugs through the gut epithelium into circulation. That is not true for all kinds of cancer therapies, right? And so what we're currently stuck at right now is we don't know, we don't have a good heuristic for what to expect in terms of how the plasma microbiome is going to change with respect to any given therapy. We're just kind of at the point right now that based on preliminary data, it does change. Great question.
Great, thanks. Next question, please.
Mark Yarshwin from Hopkins. This is a question mostly for Katie, although, interested to hear everyone's thoughts. It seems like every month that I open Science or Nature, there's some new strain of bacteria that in some model seems to be beneficial for immunotherapy. But, I sort of struggle conceptually because we know that immune responses are mostly against neoantigens, which are different in each individual tumor. So it just seems conceptually hard to imagine that a single strain of bacteria is going to be beneficial for many different patients. So I just wanted to ask, what your thoughts are about this and, whether we should look a little more skeptically at some of these papers.
I think the data are highly heterogeneous, and as mentioned, the top hits of taxa, even in one melanoma cohort, aren't really replicated in others, with a couple exceptions. And so I think it's certainly context dependent, and there are a huge number of covariates that we haven't delved into, from region of the world to race, ethnicity to liver dysfunction and other comorbidities, which probably play a huge role. I think there are some emerging themes, though, from the animal models that study these findings, that there is potential impact. And, to your question of mechanism, I think Greg wrote a fantastic encyclopedic review in Science last year, which covers a lot of the concepts, one of them being molecular mimicry, that potential that could be both bad or good in terms of a cancer response, either tolerogenic or immunogenic. But I think, I guess, and trying to put all this together myself, I think of it sort of like genomics 20 years ago, where we had just KRAS or just one or two genes, and now we're trying to put it all together into expression signatures and trying to glean a huge pattern when there's a lot of inputs and not knowing yet what's a bystander or not. But I probably should defer to Tim and Greg to answer that question, because they're going to be much better than I am at synthesizing.
The only thing that I'll add real briefly is regarding the intratumoral microbiome. So there was a really beautiful paper by Yardenia Samuel's lab at the Weizmann in Nature last March in 2021, where they specifically looked at the contribution of intracellular, intratumoral microbiota in metastatic melanoma samples. And what was really fascinating is that they identified bacterial peptides presented on MHC1 and 2 molecules on both cancer cells and immune cells. And by taking T-infiltrating lymphocytes from these same patients and co-culturing them with these bacterial peptides that were identified in these same patients led to the release of interferon gamma, suggesting that these T cells within the own patient's tumor microenvironment were actually recognizing the fact that these bacterial epitopes, per se, were being expressed on the surface of cancer cells. And so that may suggest one reason one mechanism of how these intratumoral microbes can affect immunotherapy response, at least in the context of melanoma.
That's great. Thanks very much. Next question.
I want to thank all three of you for this excellent panel. The reason that we did this was largely because of Tim's paper and putting microbiome on the map for cholangiocarcinoma and putting it on all of our minds. And Katie, even though you're not a microbiome expert, you gave us the learner's perspective, which is bringing us all up to speed on the microbiome, and it was a fantastic talk. Thank you. And we're really appreciative to have the experts of Greg and Tim on the microbiome. And Greg, we just talked to him 2 days ago to invite him because our speaker fell through. And at the last minute, he said yes and got on a plane, so we're very grateful for that. So I have two questions. One is, Greg, you made this point that maybe we shouldn't be giving systemic antibiotics because it wipes out some bacteria that could be helpful to us. And I know we have some companies here that are delivering intra-arterial therapies for the treatment of cholangiocarcinoma and hepatocellular carcinoma that are like radioisotopes, Y90, things like this. And I'm wondering if anyone on the panel or Greg, if you know if there are any therapeutic interventions that are local to try to give intra-arterial antibiotics or injecting antibiotics into tumors. And then my second question is, Tim, you might have the most experience on this panel of actually trying to collect stool samples in clinic. And I was very convinced by Katie's talk that we should all think about now prospectively looking at the microbiota. And I know we had done an IST at MGH for immunotherapy, and we tried to collect prospective fecal samples, and patients were very kindly game to help with it. There are a lot of challenges around doing it, and I was wondering if you could share some of your experience so we can all think about introducing that into the studies that we plan going forward.
Both of those are excellent questions. I'll try to tackle the first one. So the current armamentarium of antibiotics that are available for most patients are sledgehammers. As in, when you swing them, you're likely to break something else in the process. They're usually not specific. And when we think of specific antibiotics, we usually think of antibiotics that target a class of bacteria rather than individual strains of bacteria. There are some really innovative therapies coming out around phage therapy that can target some very specific forms of bacteria, and those advances may be useful in the case of trying to target those within the tumor microenvironment. Now, in terms of local injection, the closest that I've seen so far, and I have not read every paper in the literature, is that they're mostly restricted to mouse studies, and there does appear to be some benefit to local injection while avoiding systemic dosage of those antibiotics. I think a common trend that I see, again, it's a trend, it doesn't mean it's always accurate, is that getting rid of some of the bacteria in the tumor microenvironment is usually helpful for patient survival, whereas getting rid of those in the gut microbiome is often harmful rather than helpful. There's lots of ways to envision how you might get local administration. Arterial injection may be one of them. I think coupling innovative forms of antibiotics such that they're organ specific, and there are various chemistry ways to do that I don't think have been either invented or widely distributed yet, are possible, and there may be utility in exploring those methods in order to selectively eliminate microbes from specific organs that are harboring cancers.
So let me just add that William Colley injected bacteria 1860 or something into tumors. So, it's not that novel. And so we know this for a long period of time. The reason why I showed this movie is because we don't get enough samples. I think it's very important for caregivers, nurses, physicians, and patients to understand this, that, there is a significant impact, potential impact. But it's very difficult for me, and I'm sure that a lot of my colleagues, agree on this. It's much easier to put the patient to sleep or give him anesthesia, take a big needle, and get a tumor biopsy, because the patient is laying there and he has no chance other than giving the biopsy. But giving a simple stool sample is very, very difficult, and that's exactly, yes, I completely agree. It's very, very challenging, and that's why I thought I'm trying to break the barriers a little bit, but showing children's books.
If I could just interject as a clinician who tries to collect stool samples from my patients, also for Tim's study in particular, I think that this is something where a little creative shipping and collection kit resource of funds would be very helpful, because the problem is you go to your doctor at noon and you may not go to the bathroom around, in a 1 hour window before or after, and most people can't go to the bathroom on command when they go to the doctor, and perhaps it's counter to that impulse. So I think if we had invested in some good collection kits and mailers so that people could collect at home and mail it to the lab, that would not only cause a huge relief for patients and make it much easier, but also would spare a lot of the barriers on the institution side, because it's also difficult for labs that aren't used to handling fecal matter to process and package.
I think we'll
Thank
take one last question here. I don't know if there are any questions in the chat.
I want to ask about the mice that are used for cancer research. Just about all of the cures that we're finding come initially out of mouse experiments where the mouse survives, or he doesn't, he survives better, he survives worse. And these mice all have some kind of microbiome. And what we're learning today is that that microbiome has a big influence, can have a big influence on the results that we get. So we can have a situation where a researcher on the East Coast who buys his mice from the local East Coast mouse shop gets one set of results in his experiments. And another researcher on the West Coast, or certainly a researcher in Thailand, gets his mice from an entirely different shop. And his mice have an entirely different microbiome because they've been fed differently, they've been housed differently. So the results can be entirely different. And if the first result happens to be mice whose microbiome resists the treatment, we might be very discouraged by the results. The other shop might give very encouraging results. So the question is, should the results that are reported, or should the mice that are delivered, include information about their microbiome? And should this information be reported, or is it? Should there be standards for feeding the mice so that you can buy mice with this microbiome and that microbiome and understand what it is that you're getting? Is this something that is in the conversation? Should it be in the conversation?
All right, so you just lost your coffee break. Because this is very important, and there's a number of questions included, and I'm happy to answer them because it's way more interesting than you think. So we started this by indeed having, before we even thought about microbiome, having investigators, for some reason they change the institution, get a better job, and all of a sudden the experiments don't work anymore because just they take their mouse and put them in a different facility, which is exactly what you say. But I would like to mention that even if we go into patients and do those studies that Katie presented in HCC, we know that the microbiome of an Asian population is very different from a patient that lives here. So it's not. But let's go back to the mice. Yes, you're correct. So first of all, the data is all reported, so we know this. Second of all, when we talk about therapies, and there's a lot of studies where we actually use germ-free mice. So we start with 0, and then we colonize them with a set of 15, 20 different bacteria so that we really know what we have, and we can really reproduce this. But the most important is this. And this is a study that comes from my colleague, Barbara Rehermann, who also happens to work at the NIH, and she did the following, and this is why you're losing your coffee break, because it takes a little bit. She started collecting wild mice from horse farms, stables outside the NIH. And you can see exactly where those farms, where these mice were basically caught, and they collected the stool samples. And then they used these stool samples from what they call from wild mice and put these stool samples into our lab mice. And they call these mice wildlings. Okay, so now they actually have the stool from the wild mouse and not this artificial stool that we have in a laboratory condition. And yes, indeed, the results that they obtained were very, very different. And so it's not only that you see different compositions of immune cells, but it is actually so critical, and I'm just going to make it short because I know I spent too much time. There was a clinical trial that some of you may remember that was done about 15 years ago, where they used an antibody for CD28 and tested this in healthy volunteers in England. And it had detrimental effects. Some of the volunteers actually died of this. And there was no reason to understand why they died. All the preclinical work, everything was done correct. It was completely unpredictable. She repeated the experiment. Nothing was seen, obviously, in preclinical animal models. She repeated these studies now in these wildling mice, and she could actually reproduce the cytokine storm that led to the death of healthy volunteers. So, I think this is one example that shows you, number one, that, the microbiome is important, and it addresses your question of which one is the right microbiome to test.
That's great. Really a very interesting session. We're going to have a quick 10-minute break, so please grab refreshments and come back to the next session. And I think you can understand why they asked a gastroenterologist to moderate the session.